Okay, I think we have 35 should be good enough, and hopefully there are more people will join us today. So welcome everyone to join us for the bioinformatics and genomics seminar today. So I'm the senior bioinformatics scientist from the research computing department, and today Dr. Annie Adana from BCH and I will co-host this seminar. And before I actually hand it to Annie, so I would like to introduce a very useful resource for all the audience here. And I know that I actually introduced this resource for multiple times. So if you are old audience, so bear with me. If you are new audience, I would like to introduce this useful resource here. So we actually record all the seminars in YouTube. So if you are interested in any previous seminars, you can basically go to this digital science education webpage to watch the YouTube. If you scroll down, that's what the YouTube videos we have for what the previous seminars. And we also have the schedules for upcoming seminars. And so if you are interested, you can go to this page. I think that's what I have. And so Annie, can you introduce our speaker today? Thank you.
Absolutely. So I have the pleasure of introducing one of my favorite colleagues and collaborator, Nicola Whiffin. Nikki did her PhD training at the Institute of Cancer Research and then her postdoctoral training at the MRC in London, where she developed a method that was really important for variant interpretation. It was a statistical approach that used reference databases for frequency filtering for rare disease analysis, so we could figure out how common is too common. She then was a research fellow at Imperial College, where she was interested in the role of non-coding variation in human disease. And she is the recent recipient of the competitive Sir Henry Dale Fellowship and has actually just started her independent research group at the Wellcome Center for Human Genetics at the University of Oxford. She mentioned this is the 1-week anniversary of her lab, so we're all very happy for her. Her work is absolutely one to watch, both based on her success to date and her interest in non-coding variation in human disease, which is an area where I personally, and I think most of us, struggle in interpretation of genetic data. Today, she's going to be telling us about some of her work that she did in part during a visit to the Broad Institute with our group and others, where she worked alongside the genome aggregation database team, or the NOMAD team. When she came, she was the ultimate guest. Not only did she come with the project, she made excellent progress on the project. She was so productive, though, that Daniel MacArthur asked if she would take up leadership of an unfinished project characterizing LRRK2 loss of function variants that had been a previous postdoc who had gone on to another role, had left unfinished, and so asked Nikki if she was interested in taking this on, and Nikki said no problem, and she is the only one in our group, or in our larger group and among our collaborators, to have 2 first author papers in the NOMAD package. So Nikki just gets things done, and so I'm really excited to hear about her work today. Thank you so much for coming, Nikki.
All right, all right, there we go. Can you see my screen?
Yes.
Perfect. So thank you very much for the introduction, Anne. Yes, today I'm going to talk a little bit about some work we did looking at specific variants within 5' UTRs and their role in rare human disease. So this is probably no news to you guys, but just as a broad overview. So getting a genetic diagnosis for a rare disease patient is incredibly valuable both to that patient and to their family. It'll allow us to screen additional family members to find those who are also at risk of developing disease. It can sometimes lead to actually getting us a diagnosis for the patient. It can help look for personalized treatment approaches and actually can inform on how the patient will do clinically. And our current approaches to finding these genetic diagnoses are almost exclusively focused on regions of the genome that code directly for protein. And this is a strategy that makes huge amounts of sense. It's a very small region of the genome, only about 1.5%, and variants within that have likely large effects. And very crucially, that is an effect that we can predict. So we can take any single variant, and with our beautiful amino acid code, we can say, well, what effect is that going to have on the protein? Is it going to be silent? Is it going to cause a change in amino acid, or is it going to cause a new stop codon, for example? And we don't have this luxury in the non-coding space. But the problem is that we're missing something. So, I mean, this does actually do pretty well. So looking at 1.5% of the genome, we find a diagnosis for around half of all rare disease patients. But we still have the other 50% of patients who we don't find a genetic diagnosis for. So how do we start to expand outside of this protein coding space? So 98.5% of the genome is rather a lot of genome. And actually a large proportion of this has some kind of annotation when we look in data sets such as ENCODE. And there's actually a huge amount of noise. So the majority of variants that we observe will have little, a very small or actually no effect. And we can't do genome-wide tests for rare variants as they're incredibly low power. And as I said before, that we have this beautiful amino acid code in protein coding sequence, we don't have this in the non-coding space. And it's incredibly hard to define the effect of any individual variant and also the gene on which it impacts. So my approach has been to simplify a couple of these problems by looking at UTR regions or untranslated regions. So those of you who might be unfamiliar, these are regions directly adjacent to the protein coding sequence. They flank it on the 5' and 3' ends. And these regions are part of the mRNA sequence, but they don't get translated into protein. But they're incredibly important in regulating the stability of the RNA, where it's trafficked to within the cell, and the rate at which it's translated into protein. So variants within these regions could potentially have a very large impact on one or more of these processes. And crucially, looking back to the problem we have with interpreting non-coding variants, they regulate known genes. They're literally stuck to the side of them. And also there are known functional elements within these regions that actually will allow us to begin to predict the effect of variants that occur within them. So looking at these functional elements, there's one that particularly interested me, and I hope you'll find it interesting too. And these are the upstream open reading frames, or UORFs. These are distinct elements that exist within the 5' UTR of protein coding genes. They look to all intents and purposes like a mini gene. They have a start codon and a stop codon, but they're entirely encoded within this 5' UTR, as shown by this orange graphic here. Now, why these are particularly interesting is they have very important roles in subtly regulating the translation of the protein coding sequence. So a study from 2009 by Calvo et al. Showed that naturally occurring UORFs reduce protein levels by around 30 to 80%. And these things are also, they're not rare, they actually occur upstream of around half of all known genes. So just to slide a little bit on terminology. So I've introduced you to the UORF, but actually we can also have a start codon within the UTR that doesn't have an in-frame stop codon. And these will lead to what we call OORFs, or overlapping ORFs. And these can either be out of frame with the coding sequence as shown in the second graphic, hitting a stop codon somewhere within the sequence, or they can be in frame with the coding sequence and lead to what we call a transcript elongation, stopping at the canonical stop site. And another matter of terminology, we term these AUGs that occur within 5' UTRs upstream AUGs or UAUGs. So in order to understand a little bit about how UORFs regulate translation, we need to understand about translation initiation. So the ribosome binds to the 5' cap of the RNA and it scans along until it encounters an AUG, at which point one of two things can happen. So either is shown here on the top, the large subunit of the ribosome can bind to the smaller subunit and translation of the sequence can occur. Or as shown here in the second diagram, the ribosome might actually bypass this upstream AUG in a process known as leaky scanning and not translate the UORF at all. And the chance of the ribosome actually stopping at this upstream AUG is highly dependent on the strength of match of, of the surrounding sequence to the so-called Kozak consensus shown here at the bottom, with two very important positions being this -3 position being an A or a G, or the +4 position being a G. Once the UORF has been translated, if it is translated, then again one of two things might happen. Shown here on the top, the small subunit might remain bound to the DNA with the large subunit dissociating, which means the small subunit can continue scanning and reinitiation can occur at the coding sequence. Alternatively, when the ribosome finishes translating the UORF, it might completely dissociate and there'd be no translation of the downstream coding sequence. So it's these processes of leaky scanning, reinitiation, dissociation, which allows these upstream open reading frames to subtly regulate the amount of translation of the protein coding sequence. There are also two minor mechanisms that might also contribute to UORFs acting on translation. One is that this is a peptide, well, this is a sequence that gets translated, so it could be translated into a small peptide, which can function to either up or down regulate translation of the coding sequence. Or alternatively, the presence of a stop codon actually upstream of an intron-extron junction might elicit nonsense-mediated decay of the entire RNA transcript. So there have been various examples of mutations that either create or disrupt upstream open reading frames, causing disease. However, in the most part, these are single case disorders, single gene disorders and single cases. So we set out to systematically look at the effect of creating an upstream AEG. So creating a start codon within this viprom-ETR, and the effect that that has on translation and in rare disease. And this was published earlier this year in Nature Communications. So a little bit on the methods. So to look at these, we used the NOMAD database. So we used the version 2.1 dataset, and specifically the whole genome sequences within this, so we could profile the entire viprom-ETRs. So this comprised of 15,708 whole genome sequenced individuals. We use this data set to calculate a metric that we call a MAPS metric. And this stands for the mutability adjusted proportion of singletons. Now, I'm not going to go into the complex methods of how we calculate this, but I'm going to tell you why we're interested in this metric. So firstly, looking at the second part, this proportion of singletons. So we know that deleterious variants are subject to negative selection. And this action means that they are not allowed to become common in the general population. So if we look across a whole series of deleterious variants, we see they have a very skewed allele frequency spectrum, where the majority of them occur as very rare. And by a singleton, we mean a variant that occurs once and only once within the data set. So it occurs in one individual out of these 15,708. And this is purely a measure of rarity. So we're saying that they're very rare if they only occur as singletons. The second part of this metric is this mutability adjusted part. And this is because not all variants are created equally. So the chance of any variant occurring is highly dependent on the surrounding sequence context. So we take into account the base immediately preceding and immediately after the variant in question. We also know that the methylation of that base affects whether it is mutable or not. So for example, we know that C to T transitions at CpG dinucleotides are the most common variant across the entire genome. And that variant is hugely more likely to occur if the cytosine is methylated. And finally, we also need to take into account coverage, because the chance of us actually observing a variant in a data set is obviously reliant on the sequence coverage. So this mutability adjustment allows us to calculate across a set of variants, the number of them that we expect to occur as singletons. And we simply compare this to what we observe in the whole data set to calculate our MAPS metric. So don't worry if you didn't understand any of that. It's much easier if we just plot out these scores and show you what they look like. So here I'm showing you this MAPS score up the y-axis of this plot, where the higher we get up, the more variants occur as singletons, the increasing signal of negative selection against a set of variants, and we infer this as being that variant set being more deleterious. So if we plot our known coding variant types, so this model is calibrated to synonymous variants, they occur here at 0. Missense variants have higher MAP scores, so are more deleterious, and predicted loss of function variants are more deleterious still. If we actually plot all variants that occur across all viprom-ETRs, we see these are completely indistinguishable from synonymous variants. And this makes some sort of sense. With synonymous variants, we know that a large majority of them don't have any effect, but there are some that have very large effects. Maybe they impact splicing. And the same could be probably said for the UTRs. A lot of them will have no effect, but some of them may have a severe effect on translation. So if we then look at specifically these variants of interest that create these upstream AEGs, we see they're significantly more negative selection than all viprom-ETR variants. And this plot on the right here is just showing you the average number of UAEG variants that we see per gene, just to get across the fact that we're not looking at huge variants per gene here. So we start to lose a bit of power as we go through these analyses. So then going back to near the start of the presentation, I told you about the different types of upstream AEG of UORFs, where you have either a distinct UORF or these overlapping ORFs where there is not a stop codon. And we find that there is a much higher signal of selection against UAEG creating variants that create these overlapping ORFs when compared to ones that create distinct UORFs. And this makes lots of sense. So ribosome translating an overlapping ORF actually cannot translate the coding sequence because there is not time for it to dissociate and reinitiate. We also see an effect of the distance of the UAEG creating variant to the coding sequence, where variants that are created closer to the coding sequence are more deleterious. However, this might just reflect a higher propensity of these variants to create these overlapping ORFs. I also told you about the Kozak sequence and the fact that the strength of this, the surrounding sequence surrounding the ATG dictates the likelihood that translation will start at that particular start codon. I said before that there were two important bases, and here we're coding a Kozak sequence as strong if we match both of these positions, moderate if we match only one of those positions, or weak if it matches at neither. And here we see a trend that is suggestive of higher deleteriousness the stronger the Kozak, but this is far from significant. And as I said, some of these analyses suffer with power. So given that creating these upstream AEGs we expect would decrease translation, then would we actually expect them to be more deleterious upstream of genes that are haploinsufficient or known loss of function causes of disease? So to have a look into this question, we used coding constraint data created by Konrad Korczewski at the Broad Institute, based on a previous model from Caitlin Samoka's work. So here we can rank, so using a very similar methodology to what underlies our mutation correction in the MAPS score, and we can calculate for every gene and every variant category the expected number of variants we expect to observe in NOMAD, and we can compare that to what we actually observe. And here we're plotting this expected against this observed. And if we look at synonymous, so predicted silent mutations, we see that every gene, which is a different point here, lies very closely along the XY line, where they have a very similar observed number to what we expect given the model. If we look at missense variants, that we see some genes that have what we call constraint against missense variants that start to fall below the line. But if we look specifically at predicted loss of function variants, we see much higher levels of constraint where most genes show some constraint against predicted loss of function variants. And here, so Konrad Korczewski's created a score called the LERF score, or the loss of function observed over expected upper bound fraction is what it stands for, and we can rank all genes on the spectrum of this observed over expected value. And from this, we can identify two different categories of genes, those that are most intolerant to loss of function and those that are most tolerant. And here, for practical reasons, we took the top and bottom sextiles of this distribution to give us a reasonable amount of genes in each gene set. So what we can then do is we can look specifically only at upstream ATG creating variants that occur upstream of genes that are intolerant or tolerant to loss of function mutations. And here we can see this large split, and that these variants are much more deleterious when they occur upstream of loss of function intolerant genes. So a lovely reviewer of the paper when we sent it in said, well, that's great, but does this just reflect the fact that there is higher conservation in UTRs of loss of function intolerant genes? So here is a plot where we were trying to look into this. So at the y-axis here, we have the proportion of bases in each fibrom UTR with phylop scores of greater than 2. So ones that are reasonably constrained. And on the bottom, we have genes ranked in these sextiles of tolerance to loss of function, where the further to the right we go along the plot, the more intolerant the gene is to loss of function. And in gray here, we're plotting all UTR bases in these genes, and in black, we're plotting these UAEG creating variants. And what you can see is that indeed, the signal of conservation in these UTRs does increase as we move towards the right of the plot. However, the difference between the gray and the black plots also does increase as we go towards the right, showing this is not just the signal of this increased conservation that we are seeing. So going back to our MAPS plot, we can then look specifically at genes that create these overlapping ORFs and see this much greater signal of selection when we, again, we subset to just these loss of function intolerant genes. And we can then begin to try to look at known disease gene sets. However, as I said earlier, we're looking at very small, we're getting smaller and smaller variant numbers here, so the power of this analysis isn't great. So we looked at variants that are created upstream of genes that are known to be haploinsufficient, curated by the ClinGen team. And we do see this signal of selection, but again, we have massively wide confidence intervals here because we only have 137 variants observed in NOMAD in this set. We then looked at genes in the the developmental disorders gene to phenotype, or DDG2P dataset. And so these are genes that are involved in developmental disorders. And we see that across all of these genes, when we look at these upstream AUG creating variants, this is not distinguishable from all AUG creating variants in all genes. But if we specifically look at genes with a dominant loss of function disease mechanism, then we see this massively increased signal of selection. So again, this is all feeding into information that this is probably a loss of function mechanism that these variants are acting via. So hopefully I convinced you in some of that data that these variants are more deleterious when they create these overlapping ORFs. So we then thought, well, what happens if we remove the stop site of an existing UORF that is already translated? And again, this can have two different effects. Either it can create an elongated UORF if there's a stop codon further downstream in frame in the fibrin UTR, or it can create one of these overlapping ORFs, either out of frame or one of these in-frame transcript elongations. So again, back to our wonderful MAPS plot, whereas here I've switched those UAUG creating variants for stop removing variants. And we have a significantly greater selection against these stop removing variants when compared to all UTR variants. And again, we see this split between loss of function tolerant and intolerant genes, but this is no longer significant because we've reduced our variant numbers here. There are very few stop sites, and they're only 3 codons long, so we've got much smaller search space when looking at these variants. But we do see some good trends. So we compared all of these possible UORFs with a data set of ribosome profiling data, or RiboSeq data. And this is data where they pull down RNA that is bound to ribosomes and find signals of UORFs or other regions that are actively translated. And here we're splitting this by whether or not the UORF has evidence in this ribosome profiling data set of being translated. And as we expect, we see that there's larger negative selection against knocking out the stop codon of a UORF that actually is translated. Again, we see that there is greater selection against variants that create these overlapping ORFs. And here we have a significant result that this knocking out the stop codon is more deleterious when the strength of the Kozak consensus of that UORF is strong or moderate. So given that we have very few of these stop creating mutations and we have not the best amount of power to look at this, we turn to conservation as a complementary metric. And here again, I'm showing up the y-axis this proportion of bases that have a phylop score of greater than or equal to 2. And here I'm looking specifically at the conservation of UORF stop sites. So the UORF stop sites are more conserved than fibrin UTR variants as a whole. And these are matched UTR bases, which is the 3 base pairs immediately upstream and downstream of this stop site. There is greater conservation of stop sites where the UORF has evidence of translation. And where knocking it out would form one of these overlapping ORFs, either in frame or out of frame. And here again, we see this Kozak signal where there is greater conservation of the stop sites where the start site of the UORF is in a strong or moderate Kozak consensus. And actually, interestingly here, it doesn't seem to matter, and this seems to be consistent across the data, that it doesn't seem to matter if it's strong or moderate, it's just that has a signal over weak. So again, we can look at disease genes and we see that the stop sites of UORFs in haploinsufficient or dominant loss of function disease genes are more conserved than those in all disease genes in the DDG2P dataset. So we then wanted to look at kind of what is already known about these variant types in disease. So we mined both ClinVar and HGMD for all possible variants that would create these upstream AUGs and that would remove the stop sites. And we found 39 UAUG creating variants and 4 stop removing variants. And here I'm just showing you for these 39 AUG creating variants, the proportion that falls into each of these different classes. So in red here, we have these HGMD and ClinVar variants, and in gray, we have all other possible variants in the same genes. And we see that these variants that were found in cases that potentially cause disease are enriched for moderate and strong Kozak consensuses, enriched for these outer frame overlapping ORF creating variants, and are enriched for being very, close to the coding sequence. So this analysis of ClinVar and HGMD alerted us to the fact that for some genes, there seems to be a recurrently identified mechanism. And a lot of this might be due to the fact that for some genes, UTRs are more routinely screened, and this is probably enriched for things like single gene disorders. But it gave us some nice examples. And one of these is NF1, which is a gene that causes neurofibromatosis type 1. And here I'm just showing a schematic of the 5' UTR that has this one existing UORF far upstream that has evidence of being translated. And zooming into this UORF sequence, I'm showing you 4 different mutations that were identified in the 5' UTR of NF1 by a paper called by Evans et al. I think it was in 2016. 2 of these mutations, identified here in red, create new start codons, so they create new AUGs with a strong and a moderate Kozak consensus, shown by the S and M in brackets. And 2 of these variants disrupt the stop codon of this existing UORF. But all of these variants have the same final effect in that they all result in an overlapping outer frame UORF in NF1. And actually, these are the only 5' UTR variants that were identified in this cohort of patients. I think there were around 280 patients. So if you, so given that we know that these variants all have the same downstream effect, it might be prudent if you had a cohort of patients with NF1 that you would not just screen for these 4 previously identified variants, you would also screen for all of these other possible variants that would be predicted to have the same impact. And here I'm just showing single nucleotide variants that would have this impact. There are actually far more indels and frameshift mutations that would also result in these overlapping ORFs. So this also seems to be a recurrent mechanism in the previously identified variants for the gene IRF6 that causes van der Waals syndrome, where there were 3 previously identified mutations shown here on the top in red. So given that we can identify some genes where there seems to be a recurrent mechanism, we decided to rank every gene in the genome on a scale of how likely it is that UORF perturbing variants might be important in disease. So initially, we tried to characterize these genes into 9 different categories there are in total. So these 2 in dark gray are null or kind of unknown categories, where there's either no annotated UTR for the canonical transcript for some reason, or there are no possible UORF perturbing SMBs in that transcript. In gray here, lighter gray here, are the genes where we think there is a low likelihood that UORF creating variants are likely to be important. And these are genes where there are no high impact UORF perturbing SMBs possible. There is already an overlapping ORF in the gene. There is a common-ish variant in NOMAD that would lead to an overlapping ORF, or that gene is tolerant to loss of function. So we wouldn't expect knocking it down by creating a UORF to actually cause disease. On the other end of the spectrum in red are the genes where we think there is a high likelihood that UORF perturbing variants may have an impact. And these are genes that are either intolerant to loss of function or haploinsufficient and dominant loss of function genes. And finally, we have this category in the middle, which is everything that doesn't fit elsewhere. And if we collapse these into these 3 categories corresponding to low, moderate, and high likelihoods, we can then see that these HGMD and ClinVar variants that I showed you earlier are predominantly identified in these genes where we classify them as having a high likelihood of this being an important mechanism, which is reassuring. So if we then look specifically at, people tend to like gene names and looking for their favorite gene in data. So I thought I'd plot here all the genes with greater than 20 possible high impact UORF perturbing SMBs. So by high impact, I mean ones that have a moderate or strong Kozak consensus and that are predicted to form these overlapping ORFs. So these are all the genes that have greater than 20 predicted high impact SMBs. And as I said earlier, this is simplifying things because there are infinite numbers of indels that can also have this effect. But what this plot does begin to show us is why we might have observed NF1 as having this, this mechanism recurrently, and that it is in one of these genes that has the most possible mutations that would have this impact. And also it is a single gene disorder. So the people that identify those mutations in NF1 are based in Manchester. It's Gareth Evans and his team. And as I said, that paper was from a few years ago. So I got in touch with Gareth and said, have you screened any more patients? Have you found any more 5' UTR variants? Interested to see if there would be any more of these predicted variants with the same effect. And he said, well, no, but we have seen an interesting variant in NF2, and we can't predict what it's doing. And this was a variant that perplexed me for a small while, but actually now I think it's really interesting. So again, I'm showing you here a schematic of the fibrometeor of NF2. And once again, it has this predicted UORF that is far upstream of the coding sequence. And the variant that occurs in patients, so it's in 2 different families and 5 affected individuals, is a T insertion within the sequence of this UORF. And the reason this initially perplexed me is that this creates an upstream AUG, but this would create a UORF that would be predicted to terminate at exactly the same stop codon as the existing AUG, existing UORF, sorry. So we wouldn't expect it to have a large effect. But the cool thing about this is what it's actually doing is it's shifting the frame of this existing UORF that we know to be translated and transforming it into one of these overlapping out of frame UORFs. So we set about doing some functional validation work on this variant to kind of prove that our hypothesis on how this was working was correct. And this has been done by a hugely talented PhD student in the lab called Nicola Whiffin. And here we have a vector where we can clone the fibrometeor sequence of a gene of interest, either wild type or variant, upstream of a luciferase reporter gene. That luciferase readout allows us to look at the difference in protein expression with the different fibrometeors. And this is the results for these variants. So first of all, we looked at the wild type and we normalized the luciferase expression to the wild type here on the left. And then we looked at this patient mutation. And as you can see, this dramatically decreases the amount of protein expression from this allele. So in order to try and prove that it is this overlapping ORF that is causing this effect, we also, in tandem with this patient mutation, knocked out the start site of this existing UORF. And here we saw a massive upregulation of translation, or at least protein expression, to over and above the level of wild type, which is showing us that this UORF is doing some level of regulation of translation in real life. And then if we knock it out, then we see this upregulation of translation. So that's pretty neat. So hopefully I've convinced you that these variants are cool and you're excited about trying to find them in your own patient data sets. And we've done some work to try and help you do just that. So another very talented PhD student, Xiaolei, has created a plugin tool to the variant ensemble, the ensemble variant effect predictor that will allow you to identify and annotate these high impact variants within the fibrometeor. And this work is now bioRxiv, and in the paper there is a link to the GitHub repo with the tool. I thought that was easier than actually putting the long link on the slide. So this tool not only annotates these upstream AUG creating variants and stop removing variants, it also annotates variants that remove start sites, create frame shifts within UORFs, and insert early stop codons. It annotates each of these with so it annotates every variant for every possible one of these effects, and it annotates them with all of the different metrics that I've spoken about so far. So distance to coding sequence, the strength of the Kozak consensus, the effect, so whether it creates a UORF or an ORF, etc. And so far, Xiaolei has run this across all of the variants in ClinVar. And interestingly, it actually annotates nearly a third of all likely pathogenic and pathogenic fibrometeor variants in ClinVar, showing us that a large proportion of variants that we have identified in the fibrometeor potentially are having this effect. Or this might be a bit circular because there were previous examples of this effect that might have led to these things being called pathogenic, likely pathogenic in the past, if that makes sense. But what this also is telling us is that there is a large proportion of the fibrometeor variants that are called likely pathogenic or pathogenic that we don't currently know potentially the effect, how they're leading to that effect. So there's a lot more to be discovered in this space. The tool also allowed us to prioritize 31 different variants of uncertain significance that would be predicted to have a high impact effect. So these are these strong moderate Kozak consensus in one of these high likelihood genes and that form these overlapping ORFs. And our plan is to continue to update this tool as we learn more about different fibrometeor variants and the impact they have in disease. So a little about ongoing work in this space. So this paper was on bioRxiv for quite a long time before it came out in Nature Communications, organizing the whole package of Nomad papers. Took a reasonable length of time. So we started to do some work on some different data sets in this space. So one of them is looking at the Deciphering Developmental Disorders, or DDD data set, which was created by a lot of the guys at the Sanger Institute in Cambridge in the UK. And we've looked at specifically de novo variants in these cases and found quite a few that create these upstream AUGs. And I'm specifically at the moment interested in this gene called MEF2C, where we find multiple different mutations that create upstream AUGs in patients. And possibly interesting to me, because they've got an interesting mechanism, are these two that I'm showing on the schematic here. And these create transcript elongations, but they add only a very small amount to the start of MEF2C, 3 and 9 amino acids for the two variants. But they do have additional evidence. They are recurrent de novo variants, and so they each occur in 3 different patients. So there is a high prior on these variants that they are disease causing. And there are no variants like them in Nomad. And what we predict is happening here is at the very end terminus of MEF2C is the DNA binding domain. And we predict that adding the sequence to the N-terminal end is actually disrupting the binding of this transcription factor to DNA so that it cannot actually function. So we currently have lab validations ongoing to prove this effect. We're also actively running the VEP plugin across the whole of Nomad to identify all these variants with all these different effects, and on the Genomics England 100,000 genomes dataset to fully characterize which variants we see in rare disease cases. Okay, great. So in conclusion, UOFs are very important cis regulators of translation, and variants that create or disrupt UOFs, particularly those that create these overlapping ORFs, are very highly selected against. It appears that this might be an important mechanism for some genes and diseases, but there has to be a massive caveat on this. Confident interpretation of of these variants is tricky. So any variant within a 5' UTR sequence acts actually in combination with all the functional elements that are within that UTR, and every UTR is different. So actually it's not as simple as it is in the coding sequence, where it's actually not that simple a lot of the time anyway. But validation of these variants is going to be incredibly important to prove that they actually cause disease in patients. And finally, just to say that large population data sets such as Nomad are a hugely powerful tool to actually pinpoint different non-coding variants with a predicted effect that are highly deleterious and can lead to rare disease. So that just leaves me to acknowledge my team. So my old team at Imperial College, specifically my mentor James Ware and Jale and Nick, who I mentioned for their work. The team at the Broad Institute of MIT and Harvard, which is a collaboration, so, particularly Daniel MacArthur and Konrad Kochevsky, and the people in Manchester for their help with NF2, and the people in Singapore for their help with the ribosome profiling data. Thank you.
All right, thank you so much. Very interesting talk. And we actually have several questions from my side. I'm not sure about Annie's side. So there's a first question from Wenhan Tan. So Wenhan Tan, can you actually unmute yourself and ask the question?
Yeah, so my question is, for a given gene, how do we know whether the YOF is YOFs would be important, variants in YOFs would be important? So like looking at the graph that you showed, it was very interesting. You know, P10 was right up on the intercept of the x-axis. And we do know, for example, there are a lot of upstream variants in P10 that are pathogenic. But my question really is, given, you know, I'm looking at variants in given gene, I'm worried about given gene, how do we know whether the YOFs is important? Is the Ensemble VEP plugin the way for to look at it?
So as I said, yeah, this is a really important question, and it's really important, I think, to study individual genes. So I showed you a really, really simplified picture of how UORFs exist, and that was a single UORF in a single gene. And that is often not the case. And P10 is a cancer gene, right? And actually tumor suppressors and oncogenes tend to have huge UTRs, which have multiple UORFs in tandem. And there seems to be some sort of redundancy. So actually predicting the effect of knocking out a single stop codon in a UTR where there are multiple UORFs is much more complicated. So actually there is a need for functional validation. And actually I think it's going to be really interesting. There are some plans, various different groups, to do kind of systematic mutagenesis approaches across UTRs. And I think that kind of data is going to start to show us how these different elements interact in terms of translation. Does that sort of answer your question?
Sort of. I mean, I guess from a gene discovery or just from a practical perspective, you know, you've done exome sequencing on a kid or patient. You're worried that this patient may have a mutation in a given gene, but you have not been able to find it. And really, I think with your really nice data and talk, the question is, do we need to look for that particular gene that we're interested in? Do we need to look for the upstream ORFs, variants in upstream ORFs, or?
I would say yes. I think to me the natural next step is, if you don't find anything in the coding sequence, is to look at the UTRs, because they're highly regulatory. And I think there are no mutations in 3' UTRs that knock out poly-A signals and things like that. So there is, and I think if you find a novel variant in a UTR, it becomes in a gene that you know is a cause of that patient's disease, especially when it's a single gene disorder, or they show a phenotype that's very similar to patients with the mutations in that gene. I think it makes sense, very logical sense, to do the next step to be screening the UTRs and looking for UTR variants.
Thank you.
All right, we have second question from Benjamin Robbie.
Hi, thank you. So that was a wonderful talk. You alluded earlier about some of the UAUGs can be lost, that you've screened for those variations as well. Have you found any examples where a loss of the UAUG results in disinhibition, an upregulation of gene expression, and that might be a gain of function mutation by having an overdose of the gene target?
Yeah, so there's a couple of things I can speak to there. So firstly, one of the papers I showed on the slide where I had some papers of different mutations is a study where they looked specifically at somatic mutations upstream of, I'm going to get tumor suppressor oncogenes the wrong way around, upstream of, cancer genes that are upregulated somatically in cancer, basically. So yes, there is a proven effect that these mutations actually act via gain of function.
But that's somatic.
Second bit of data. Oh, sorry.
That's somatic. You're saying in cancer lines that's been shown.
Yes.
Yeah.
Secondly, speaking to that, is we have some in-house data. So my Imperial lab work a lot on cardiovascular disease and are interested a lot in fibrosis. And there are some of the key regulators of fibrosis have UORFs in their upstream UTR. And when you functionally knock out the UORF, you massively upregulate expression of the fibrosis regulator. So there are definitely evidence in there. I can't remember, going back to the ClinVar data, of how many there are in ClinVar that that knock out start sites. There are quite a few start loss mutations in ClinVar, the second bar here. I haven't looked into them in detail though.
Great.
Right. We have a lot of questions from Pankaja.
Yeah, thanks so much for the wonderful talk. I have a quick question about how much these splicing pipelines like Splice AI or other ones can pick up on these variants, and do we need something unique about these 5' or they can be picked up on these pipelines? Thanks so much.
So these particular mutations won't be detected by splicing algorithms because this is not a splicing mechanism. There are definitely disease-causing splicing mutations in 5' UTRs. I'm not sure about 3' UTRs. Around a third of all genes have an intron, at least one intron in their 5' UTR. So there are disease-causing splicing mutations, but the splicing algorithms won't pick up these particular mutations. Another important thing to note there is that 5' UTRs historically have been pretty badly annotated. The main project, which is kind of RefSeq and ensembles, approach at trying to define a single transcript per gene is improving our annotation of fibrillary UTRs. But sometimes the splice algorithms can fall down looking at UTRs because the introns and exons aren't actually well annotated.
But in terms of like you're talking about like a new start codon, right? AUG, so that should technically be picked up by a splice pipeline if it is the case. I'm just wondering why it's not going to be picked up.
I don't 100% understand. So a new start. So I don't think splice algorithms detect
a new start codon, like for example. I'm just curious. I don't know myself. So
I didn't think they do.
I didn't think they did either.
Okay.
Because they're trained on the splice consensus sequences and some of the surrounding variation, but they're not trained to pick up start sites.
Okay. Thanks.
All right, I have one question from my side. It seems like we'll just kind of stop removing and AUG creating variants are very important. So and then you create this tool it's called UTR annotator. So one question for the UTR annotators is this plugin accept what the VCF files, or is like for whole genome sequence, or is only for the UTR sequence variants? I mean for the input data.
So the input data is any data that you can input into VEP normally. So VCF files, we're working with the VEP team to get it working on the web browser itself and to be downloadable from their repository to make it kind of easier to find and use. At the moment, it's separately downloaded from our repository. Yeah.
I see. So do you have do you have any plan to create this kind of non-coding variants database? So
I don't have a particular plan. There have been some papers in the past that have tried to collate all known non-coding pathogenic mutations and some sort and kind of the regions that they occur in, but I've not made any strides towards that.
I see. Yeah, thank you. Do we have any more questions? If not, Annie, do you have anything want to talk?
I mean, I've seen Nikki's amazing work many times. We've tried it in some of the Center for Mendelian Genomics data. One of the actual features from that is initially when Nikki had developed this and was running it, she was focusing on genomes because like the really good capture of the non-coding variation. So we have not tried it on our exomes, but we're very excited to, just because you actually do pick up some part of the 5' UTR often in exome data.
Yeah, that's one thing to say actually. So the DDD data, they're all exomes, they're not genomes. So there is definitely some sequence. And I think the amount that you capture is dependent on whether there's an intron or not in the 5' UTR. I'm planning to do an analysis to look at the coverage in DDD against the main transcript 5' UTRs. So I should be able to tell you at some point soon exactly how much is covered in a kind of standard exome pipeline.
All right, I don't have any question from my side anymore. So thank you so much, Nikki, and a really nice, interesting talk. And if any other people have any question, you can contact Annie, Nikki, by emails or.
Yep, feel free.
All right, thank you guys.
Right, thank you.
Thanks.